Fungi are eukaryotic achlorophyllous organisms which reproduce by asexual and sexual methods and produce spores during reproduction. Their somatic body typically remain covered by a wall which is composed of chitin, glucan, cellulose along with other macromolecules. This group of organisms represents a unique group because of their cellular organization and behavior. It has been estimated that about 1.5 million fungi inhabit the Earth's surface, of which only 5% are identified and characterized. The systematic study of fungi is about 389 years old, but their manifestation has been recorded for thousands of years. Classification of fungi is a very difficult task because of the problems arises due to the opinions of different workers, and such difficulties are mainly due to the approaches, collection of data and interpretation of data. The history of fungal classification can be traced back from the time of Bauhin, famous herbalist, who published a simple list of 100 fungi in his book Pinax Theatri Botanici in the year 1623. After that, a number of classificatory systems have been published by different workers from time to time, some of which gained importance and wide acceptance. For example, Saccardo published his system in the year 1882, Clements and Sears in the year 1931, Bassett 1950, Webster in the year 1970, Ainsworth in the year 1973, Moore 1980, Hawksworth in the year 1983, 1995, 2002, Alexopoulos, Mims and Blackwell in the year 1996, Deacon 2006, Webster and Weber 2007, Hybet et al. 2007. More recently, fungi are classified on the basis of biochemical data, genetical data, etc. But conventionally, they are classified on the basis of their traditional features utilized by the mycologist. For example, nature of their somatic structure, type of asexual spores, nature of sporangia, life cycle patterns like asexual cycle, sexual cycle, haplontic cycle with restricted dikaryon, haplontic cycle with dikaryotes, diplontic cycle, haplodiplontic cycle, dikaryotic cycle, and so on. Recently, a great deal of informations are coming out from the field of biochemistry, molecular genetics, etc. As a result, the classificatory systems is to be modified regularly in order to reflect the interrelationships, origin, and evolution of the group. In this brief resume, I will try to concentrate on two different classificatory systems, one proposed by Ainsworth in the year 1973 and another published by Deacon in his book Biology of Fungi in the year 2006. Now comes to the system proposed by Ainsworth GC in the year 1973. Ainsworth placed fungi under the name Mycota in a separate kingdom and divided the kingdom into two divisions. One is Myxomycota, another one is Eumycota or true fungi. The members of Myxomycota again divided into classes Acrasiomycetes, Hydromyxomycetes, Myxomycetes and Plasmodiophoromycetes. But the true fungi or Eumycota are divided into 5 subdivisions, namely Mastigomycotina, Zygomycotina, Ascomycotina, Basidiomycotina and Deuteromycotina. The Mastigomycotina includes 3 classes, that is Chytidriomycetes, Hypochytidriomycetes and Oomycetes. But the subdivision Zygomycotina divided into 2 classes, Zygomycetes and Trichomycetes. Ascomycotina divided into 5 classes, Hemiascomycetes, Loculoascomycetes, Plectomycetes, Labelbeniomycetes and Discomycetes. The subdivision Basidiomycotina includes only 3 classes, Teleomycetes, Hymenomycetes and Gasteromycetes. The subdivision Deuteromycotina divided into Blastomycetes, Hyphomycetes and Colomycetes. Now, let us see what is the difference between the division Myxomycota and Eumycota. The members of Myxomycota are characterized by their presence of plant body, which is plasmodial in nature or pseudoplasmodial in nature. Plasmodium is nothing but a naked multinucleate mass of protoplast which moves by amoeboid movement and exhibit phagotrophic nutrition. Now see the diagram of a plasmodium, and it is the electron micrograph of extending plasmodium and how beautifully a plasmodium looks like. Plasmodium are basically free-living holocarpic diploid structure. During reproduction, it gives rise to a beautiful fructifications or fruiting structure inside which the spores are differentiated after meiosis. Spores of Myxomycota are typically with a rigid cell wall like true fungi. Now, the characteristic features of Eumycota or true fungi. The somatic body of true fungi is mostly filamentous, highly branched, well-developed structure called hyphae. These hyphae altogether constitute what we call mycelium. This is the structure of somatic body of a Eumycota, where you can see the filament-like structures, that is the hyphae, composing what we call a mycelial mat. However, there are some true fungi which are unicellular, for example yeast or yeast-like members. Now, this hyphal body usually composed of a tubular structure which may be septate or aseptate. When septa is present, they are always provided with a simple central pore, as you can see in the diagram. Now, the cell wall of true fungi is composed of chitin, cellulose, chitosan, and glucan. The difference between chitin and chitosan is that chitosan is nothing but a deacetylated form of this chitin. Asexual reproduction in Eumycota takes place by means of mitospores, but sexual spores are produced after fusion between either sexual apparatus, sex organs, sex cells, or even nuclei. But the most characteristic feature and important feature of this Eumycota is that gradual and progressive simplification as well as elimination of sexual apparatus or sex organs. In the lower group of fungi, you will find that typical male sex organ Anthridium is present. Female sex organ, either Ugonium or Ascogonium, are present. But in higher fungi, say for example in Basidiomycetes, you won't get any reproductive structure like Anthridium, Ascogonium, or Ugonium. In that case, hyphal fusion leads to sexual reproduction or responsible for reproduction, where only two nuclei fuses with each other. So you can trace the gradual elimination from lower group to higher group very easily and simply. Now, the characteristics of Mastigomycotina. It is the first subdivision of true fungi, where you can see the somatic body is composed of hyphae, and which together gives rise to what we call mycelium. The hyphae is sinusoidal, very tubular, branch structure that is devoid of any cross wall or septa in between. So, sinusoidal hyphae represents a multinucleate structure, tubular structure. Now, the cell wall composition of Mastigomycotina differs from one class to another, and that characteristic feature is very important because one class consists of one type of chemicals and naturally differs from the other. For example, Chytridiomycetes consists of chitin and glucan in their cell wall. Hypochytridiomycetes consists of cellulose and chitin in their wall. But the group Eumycetes is devoid of chitin in their cell wall, and naturally their cell wall composed of only glucan and cellulose. As you can see that in Chytridiomycetes, chitin is present, cellulose is absent, glucan is present. In Hypochytridiomycetes, chitin, cellulose, and glucans all are present, but in Eumycetes, chitin is totally absent. Sometimes these chemical characteristics help the mycologist to solve morphogenetic problems. Now, during asexual reproduction, mitospores are produced which are motile in nature, that is equipped with flagella, or what we call reproduce asexually by means of planospores, that means motile spores. Now, in holocarpic form, the entire content of the plant body converted into the flagellated structure, that is the zoospores. But in eucarpic form, as in Phytophthora or others, after a vegetative growth, the sex structure or the asexual spore producing structure, sporangium, are differentiated. But it is to be noted that asexual spore producing structure never united to form macroscopic fructifications in this group. Three types of zoospores are observed in the life cycle of members of Mastigomycotina. They may be anteriorly uniflagellate. Whenever it is anteriorly uniflagellated, it is equipped with a tinsel type of flagellum, that is flagellum provided with small hair-like structure, that is mastigonyms. Second type of zoospore is posteriorly uniflagellate. Flagella is of whiplash type. And the third group represented by the biflagellate zoospore, where one flagellum is of tinsel type and another is whiplash type and may be present either laterally or anteriorly with whiplash flagella projecting backward. During sexual reproduction, usually isogamous or heterogamous reproduction is observed. In case of isogamy, two fusing gametes are of similar size and shape, but in case of anisogamy, you just see that one gamete is larger than the other. In case of oogamy, the female gamete is sluggish enough and may be confined within the gametangium, but male gametes are always smaller and motile. Now comes to the characteristic feature of Zygomycotina. The members of Zygomycotina are mostly saprophytic, maybe facultatively or weakly parasitic, but their somatic body, like Mastigomycotina, are sinusitic in nature. They are tubular, hollow structure without any cross wall in between, and thus they are multinucleate. This is the somatic structure of Zygomycotina, which is tubular, sinusitic, without any cross wall. However, in older hyphae, one can find solid septa in between. These solid septa are produced just to delimit the sex organs from the vegetative body, or to block the damaged part, or to seal the mechanically damaged part of the hyphae. Otherwise, all the content present in the tubular structure or vegetative body will run out. Now, chemically, the cell wall is composed of chitosan mainly. Just few minutes ago, I mentioned that chitosan is nothing but deacetylated form of chitin. You can see from this structure. Asexual reproduction in this particular group takes place by means of mitospores, which are produced in sac-like sporangium. They are always non-motile, that is aplanospores. The sac-like sporangium may be columellate or non-columellate. Just outside the columella, one can find the sporoplasm, which before maturity of spore undergo cleavage and gives rise to numerous small non-flagellated aplanospore called sporangiospores. Sexual reproduction in Zygomycotina takes place by means of gametangial copulation, where the copulating gametangium may be similar size and shape or may be of different size and shape. In rhizopus mucor-like members, you will find two copulating gametangium of similar size and shape, and that is why it is called isogametic copulation. But in case of heterogametic one fusic gametangium is smaller than the other, which is little bit larger. This type of heterogametangial copulation is observed in Zygorhynchus like member. Whatever may be the nature of copulating gametangium or the mode of sexual reproduction, they always produce a typical sexual spore called zygospore, and from this name of the spore, the name of the group Zygomycotina derives. Zygospores are mainly thick-walled, warty, black structure inside which the diploid nuclei are present. Before germination of zygospore, meiosis takes place, and as a result, haploid spores are liberated after germination of zygospore. Before liberation of spore or before undergoing meiosis, what happened? The outer dark thick wall of the spore cracks, the inner wall protrudes in form of a small tube and gives rise to a terminal sac-like meiosporangium containing numerous meiospores. So, this type of sporangium is called meiosporangium to distinguish it from the sporangium produced during asexual reproduction. Dikaryotic phase in Zygomycotina is totally absent, and motile spores are also absent in the group Zygomycotina. In my next lecture, I will consider the characteristic features of Ascomycotina, Basidiomycotina, and Deuteromycotina.